Hello students. So in this video, we'll discuss about the DNA replication next part. So in previous video we discussed about the enzymes which are used in the replication process. There are more than 10 enzymes so we have taken. So in that video we have discussed, so that that is important, okay, and because a group of or set of catalysts, what we call a set of catalysts. So catalyst means here, so that is nothing but enzymes, okay. So enzymes which are involved in the replication process which we have discussed in previous video, right? So now let us discuss about the main theme of the replication, that is what machineries and the enzymes. Here machineries means the mechanism. So with the enzymes, how exactly the replication takes place, so in the cell. So usually in E. coli cell and in there is maybe little difference which we will see in the next video. So the set of catalysts are involved in both the E. coli as well as in the almost all living beings, right? So there are some different enzymes may be utilized, right? See, replication means, as you know, so in another name we can also say that is polymerization of deoxyribonucleotides. Yes, polymerization of deoxyribonucleotides. So actually nucleotides are the monomers of DNA, right? So that DNA is nothing but it is a polymer, right? Polymer means here polynucleotide strand, what you call polynucleotide strand, right? So that polynucleotide strand is formed by the monomers, that is called nucleotides, right? So nucleotides are present in the cell, right? Nucleotides are present in cell now, so they are joined by the help of phosphodiester bond, so which makes it a long chain, that is called polynucleotide strand. Then again the two polynucleotide strands are joined by the help of the hydrogen bond and in between what we call nitrogen bases, and makes it double stranded, and that is twisted, so which makes the double helix model as explained in earlier video, right? Double helix model, which is explained us by the first time by what Watson and Crick. So even we call it as a Watson and Crick model. So as we know about in a E. coli there are nitrogen bases. How many nitrogen bases are there in that elongated DNA? That is 4.6 into 10 raised to 6. 4.6 into 10 raised to 6 yes, base pairs, right? So this is about the 4.6 into 10 raised to 6 in E. coli, right? In E. coli bacteria, right? In case of human beings, diploid cell content may have about diploid cell, so that is 2n have about 6.6, right? 6.6 into 10 raised to 9 base pairs, right? In human cell, right? Human cell. So in each cell we may observe this much of base pairs in each DNA molecule, right? So see, the polymerization, the rate of polymerization when you compare, so this nitrogen base pairs about this, right? So 6.6 into 10 raised to 9 base pairs are joined and forms a new DNA molecule, so in 18 minutes, right? So that is 18 minutes, it will take about 18 minutes, right? So about when you calculate this one, about 2000 base pairs, what you call base pairs are joined per second, right? So per second 2000 nitrogen base pairs are joined, so they are binded each other by phosphodiester bond, and there is a with a hydrogen bonds with another strand, and makes it a double stranded DNA, right? So this is what the complex mechanism which takes place here. Let us start with this one. There are many steps, right? So and with high accuracy, so it's a plus highest accuracy, yes, high accuracy is also important. So if it's done any mistake, so in the during this base pairing, so that mistake is said to be what you call the mutation, right? So it may cause the mutation, and the more mutations cause the change in variations, right? That may cause in next generations may get the new species also, right? So DNA, as usual, you know that stable, which is very less reactive and more stable as we studied. So that's why the high accuracy is also maintained in this much of fast rate of polymerization is also so fast. 2000 base pairs are joined per second. In 1 second 2000 nitrogen bases are joined to make a strand, right? So here, for making the polymer strand, so there is a requirement of deoxyribonucleotides, right? Deoxyribonucleotides. These deoxyribonucleotides are joined, they are joined to form a polynucleotide strand, right? So now the steps involved in this mechanism of replication is, first I will take the activation of nucleotides, activation of nucleotides. So here deoxyribonucleotides we can say, so that is deoxyribonucleotides, here it is there, no? So deoxyribonucleotides, right? See, there are 4 types of nucleotides, as we know, 4 types of nucleotides. 4 types of nucleotides are, so based on types of nitrogen bases, right? There are 4 nitrogen bases, as you know, that is adenine, guanine, next cytosine, thymine, right? 2 are purines and 2 are pyrimidines, right? So these are in the nucleotide of adenine nucleotide, right? So we call it as adenosine monophosphate, guanosine monophosphate, cytidine, yes, cytidine monophosphate, and thymidine monophosphate, right? So these are the 4 types of what you call nucleotides. So even we refer it as deoxyribose, because it is we are discussing about the DNA replication. Deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, deoxythymidine monophosphate. So these monophosphates are undergo phosphorylation, right? Phosphorylation. So by the help of phosphorylase is one of the enzyme which is involved in the addition of phosphate, and it's become, so dATP, so this one is get dGTP, and this one dCTP, and this one dTTP. So these are what we called, so these are there in the inactive form, right? So these nucleotides are there in inactive form of nucleotides. Inactive nucleotides, I'll write here, inactive nucleotides undergo phosphorylation. So that is with the help of phosphorylase, an enzyme, right? So this converts it into an active form, active nucleotides, right? So these active nucleotides are having dual purpose. So dual purpose means, so they are having 2 functions here, dual purpose, they need for the dual purpose. So they are, first one, so I will take here, so it acts as a, yes, it acts as a energy liberating molecule, which is providing the energy, which is providing energy, and also it is acts as a, yes, the substrate for the polymerization. Substrate, these are 2 important concepts here. So this activated nucleotides are serving dual purpose. They are serving dual purpose, means the 2 functions are carried out by the same molecule. So that is able to provide the energy during, this is a highly energetic process because it requires the energy, right? So bonds are formed here, breaking of bonds and formation of new bonds, so it requires energy here, right? So this is one of the metabolic activity, means there should be a requirement of ATPs, right? So in the same way ATPs are serving the metabolic activities, here also this ATP is here, or GTP, CTP, TTP, what we call, so these are deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, deoxythymidine triphosphate. So these triphosphates are active form of nucleotides, and which serves the two functions here, that is one is energy, they are providing the energy, another function is substrate means nucleotides are joined to form a strand. So which is the substrate, the main molecule in the strand. So that is what the substrate we can say, right? So this is what the first step we can consider, but don't think that here the steps one after one. So it's simultaneous process. For our understanding, we are made here some steps. The second step I will take, so that is opening of helix. Opening of a double helix, or what we call helix. Of course DNA is double stranded, which will get opened. So with the help of unwindase, an enzyme unwindase, or we can say helicase, right? Here helicase is very important enzyme, helicase or unwindase. This enzyme, the unwindase or helicase enzyme, can open this double stranded what we call. So if you consider this is the double stranded DNA which is get opened, yes. So the opening of DNA is very specific, so which will actually the first recognition of ORI site. So what we call the first one is recognition of ORI site. Here again sub step you can consider. Recognition of ORI site. Recognition of ORI site means here, so where exactly there is a particular region on this elongated DNA. So there is a particular region where specific sequence are there. In that sequence it makes the broken of this hydrogen molecules, hydrogen bonds, right? Not hydrogen molecules, that is hydrogen bonds between these nucleotides, between two strands, which will be opened. Okay, so opening of the helix starts with this helicase. Okay, so that is a recognition of ORI site. ORI site means that is origin of replication site. So this is called origin of replication site, where the replication begins. So actually there are actually two kinds of we can say here, if circular DNA is there, so I'll take here, if here circular DNA, see for example here it is circular DNA in case of bacteria, right? So this is what the circular DNA. So here where what happens, one side it will get opened like this, right? So which is become like this. So this is what the so bubble like, so there is a formation of bubble like structure, so which is called the bubble replication, or that site is referred as the replication fork in case of linear DNA. So in case bubble in a circular DNA, replication bubble, so we call it as replication bubble, right? So it's not bubble like, where because open ends are not there, no? So right, so this is what the replication bubble. So in this way we can say here, so this is the open structures, right? Here it will be get double, from here it will be begins. So but in case of linear DNA, so that was there earlier like this, so then it will be opened. So when it is opened, so here, so in one end there is a formation of yes, I'll take here. So for example, this is the DNA, linear DNA. Here what happens, in this side it will be opened. So in this side it will be opened like this, right? So this is called the Y fork we can say. Y like a fork is formed, fork replication fork we can say, replication fork. In this way there is a in the ORI, this is what we call ORI, this is the ORI, this is also here ORI. So where replication is begins, right? So this is what the two steps we observed here. And next the opens up the helix by the helicase, as I mentioned here. Helicase is one of the enzyme. So where the helicase, for example, if helicase is there, helicase enzyme, so we will go on like how the opening of the zip, right? So this is what the point where it will carry its opening. It will break the hydrogen bonds and this will move like this. So it's become, I'll show here. So one more step. In this way, so it will go up and by opening that new strands. Again the new strands. So new strands are formed like this. We'll see the structure later, right? So full structure we'll see in the next step. So this is what the opening of helix, right? Opening of the helix. So even the stabilization of opened, because when these strands are get opened, so there is a tension is created. Okay, tension is exerted on this opened strands. So which are released by the what you call some kind of proteins. So there are proteins. So single strand binding proteins, what we call, these proteins are called SSBP, single strand binding proteins, which helps to release the tension which is arised in the opened strands, right? Then they are become the straight. So they may get like this. So this is get linear completely due to presence of these single strand binding proteins. So these are also involved in this one like enzymes, right? So these are the two steps. Let us take the next step in the next bit. Thank you, students.